Proteins are our fourth and final category of macromolecules. We're going to talk a little bit about something else at the end of this video, but as you can see, when you look at proteins, you see meat, you see eggs, you see beans. A lot of foods that have a lot of proteins are shown in this image. So monomers of proteins are called amino acids. Okay, here is the actual structure. You don't have to know it, but it is kind of interesting to look at. And so you're going to see a bunch of these linked together and folded and coiled and maybe even attached to another separate one. And they are going to end up something like this in a 3D rendering of in a computerized image. So amino acids also have components. They have one R group, which is any kind of functional group that changes the job of the amino acid or changes the job of the protein overall even. There's an amino group, which is where the name comes from, and then there is a carboxyl group, which is the acidic group. Okay, so you can show here amino group here, carboxyl group makes the acid, and then here is the side chain, which is the R group, and it gives it a specific job or function. Because some of these are hydrophobic, some of them are hydrophilic, some of them are acidic, some of them are basic, and there are a total of 20 kinds of amino acids that make up all proteins. So you can have any kind of length of any number of amino acids linked together, and you can have them in any organization in any amount of repetition. So there are unlimited versions of proteins. So the polymer name is called a polypeptide when it's really long. So when you connect over 100 amino acids together, you get a polypeptide. And most of the times we'll respond and call them polypeptides. And then peptides are short ones, so if they're like 10 or shorter connected together. And so a protein's structure determines its function. The law in proteins and in DNA and in central dogma is that structure creates function. Okay, so the sequence of the amino acids determines the protein's three-dimensional structure. And the three-dimensional structure is integral to its function. Okay, if it doesn't fit together with something, then it's not going to work. If it doesn't go where it's supposed to go, it doesn't work. If it doesn't fold a certain way, it doesn't work. Okay, so the structure means that it has a function. So there are 4 levels, but we're going to just kind of go over them briefly. Okay, so the primary structure is the unique sequence of amino acids. So when you link up all the amino acid monomers together, that's the primary sequence. So this just chain of amino acids is the primary structure. The secondary structure is when you take that chain that you just formed in the primary structure and you either fold it back and forth and create a whole flat beta sheet with it, or you take it and coil it kind of the way DNA looks like and you get an alpha helix. So those are 2 separate secondary structures, but keep in mind, they're both secondary structures. Now, the R groups interact with each other differently. Remember, hydrophobic ones will like hydrophobic ones. Hydrophilic ones will like hydrophilic ones. So the folding of that alpha helix is going to crinkle up based on the hydrophobics wanting to be near each other, the hydrophilics wanting to be near each other. Or the sheet is going to crinkle and fold up based on the acidic part being attracted to each other, or the basic part being attracted to each other. So that is the tertiary structure. And then quaternary means that you're going to take a second protein chain formed from the primary, secondary, tertiary, and you're actually going to take them separately, and they're going to come together and form together. Okay, so they're technically 2 separate pieces, but they will work together in their function. So that is the quaternary level of structure. It's actually technically 2 separate polypeptides. So quick check, what is the monomer of a protein? It's called an amino acid. And what is the polymer of a protein? A long one is called a polypeptide, and a short one is just called a peptide. So here are the functions of a protein. And so we have this acronym called MEANS that will help you remember it. So the cell membrane contains a lot of proteins, right? They help transport things. So they are all over the cell membrane. They make up enzymes, which we'll talk about in more detail because chemical reactions are super important for metabolism, for being alive. So proteins are the macromolecule that all enzymes are made out of. They make up antibodies that help you target foreign invaders in your immune system so that your white blood cells can go attack them. Without these tags or targets, your white blood cells do not know who to attack. They make up non-steroid hormones. There are some hormones that are not steroid or not lipid-based, and then therefore they are made of proteins. They're not made of carbs, they're not made of nucleic acids, they're either going to be made of lipids or of proteins. And then they're huge in structure. Remember what I was saying earlier, where if you don't have proteins, you don't have a physical body, you don't have a physical structure. So they are the major structural molecules in living things. Growth and repair, they make muscles, they make ligaments, they make tendons, bone, hair, skin, nails, all of the above. So again, proteins, think means. Membranes, M, enzymes, E, antibodies, A, non-steroid hormones, N, and structural molecules, S. So enzymes in a little bit more detail now. An enzyme is a protein that is made to catalyze or speed up a specific type of chemical reaction. So each kind of chemical reaction is going to have its special kind of enzyme. And that enzyme only does one job. Okay, and it does that job over and over and over and over again at high efficiency and very high accuracy. Okay, a substrate is the thing that the enzyme is going to work on. So it's kind of like the clay that it's going to remold. Okay, the product is what comes out after the reaction happens. So it's a substance formed in the chemical reaction that the enzyme was a part of facilitating. The active site is the region on the enzyme that binds to the substrate. So it's like a lock and key. If you look at the picture here, you can see that the enzyme here is in blue and the substrate is in green up the top. And they fit together like a lock and key. It's like puzzle pieces. And afterwards, when it forms the products, it's going to let them go and they will be different from the initial substrate that is coming in. Okay, substrate, enzyme, active site right here that locks and keys in with the substrate, right? It matches perfectly. And then the enzyme stays the same, is not changed because it's going to go on and do the same reaction over and over and over again until the substrates are gone and the products are going to leave. That way the active site opens up again. So how enzymes are affected are that they can be affected by temperature. The optimal temperature is going to be the temperature that the enzyme works the best at. So it's going to have the highest rate of reaction. It's going to turn the substrate into the product at the highest speed possible over and over and over again. The same goes with pH. So if something is too acidic or too basic, it's not going to work as well. But if you're at the optimal pH or the optimal acidic level, then the reaction will go as fast as possible and the enzyme will react with as many substrates as possible to make products. So the problem is when you are not at a good temperature for the enzyme to react, and it is an extreme temperature or an extreme pH, the enzyme can become permanently altered and then deformed and it can't do its job anymore because remember, structure equals function. That's called being denatured. So denaturing is destroying the structure of the enzyme to make it no longer able to perform its function. You end up with low or no rate of reaction anymore. So that's kind of extreme though. Usually if you just change the temperature or the pH a little bit, then the enzyme just doesn't function as well. And then if the pH and temperature changes back eventually, remember these are also separate. It could just be pH changing or just be temperature changing. But if they change back, then the enzyme will go back to its normal function. If you denature, it's extreme. So it's like you're going from 0 to 100 degrees. And then the enzyme's like, nah, I can't do my job here. It's going to denature. It's going to change shape and it's basically going to be able to cease functioning. If you change the pH and the enzyme is used to being in a basic alkaline environment and you change it all the way to super acidic, it's going to denature as well. And it's going to become permanently stuck in this new shape where it cannot do its job anymore. So this is kind of the picture showing that. Originally the substrate and the enzyme match perfectly, but when it's denatured, they no longer match. So the enzyme can no longer speed up the chemical reaction of changing the substrate into the product. Here's a graph that you're probably going to see a little bit where it's a curve because the enzyme doesn't just only work at one temperature. It works reasonably well at multiple temperatures here. And you can see that over here, when it's colder, the enzyme works worse and worse and worse. And over here, when it gets too hot, all of a sudden it stops working because it probably gets denatured and it can't work at all anymore after it gets beyond a certain temperature. But the highest peak is the optimum temperature because it works the highest rate right here. So it's doing the most work, the fastest, and the most efficient. So examples of proteins are animal products like dairy and meats, also enzymes like we just said. A little bit more information now. If you need to create a polymer or you need to break down a polymer, you're going to have to do one of two processes. So dehydration synthesis, if you're dehydrated, it means you're lacking water, right? It means that your water has been taken away. So when you take away water, you're going to synthesize or to make, right? The synthesize is like to make drugs or to create things. Right? Dehydration synthesis means take water away to create. And that's going to be when you take water away from a monomer, two monomers, and you're going to end up creating a polymer. Okay, so by losing the water, monomers get stuck to each other, and then you can do that over and over and over to make a chain of monomers on and on and on into a polymer. On the opposite end, if you're trying to break a polymer, you're going to do this thing called hydrolysis or hydrolysis. And lysis means to break. And we're going to talk about that more when we talk about what happens to cells when you put them into the wrong kind of environment. But hydro means water. So you're going to add water to break. So when you want to break a polymer, you're going to add water with the enzyme that's going to catalyze this reaction. And it's going to break apart the bonds in between the chains. And you're going to get monomers separated out again. So here's a picture, right? Dehydration synthesis. If you pull the OH And the H from here, you're actually going to have a bond between the carbons with the O, and you're going to have going from a monosaccharide and a monosaccharide to a disaccharide. And if you keep doing it, it's going to become a polysaccharide when you have more and more and more of these hexagons connected to each other. And then you also have a hydrolysis example where you have this disaccharide to begin with, and you're going to add water, and it's going to add back onto where this O was to the carbon, and you're going to end up with two separate monosaccharides again from this disaccharide originally. And you can do this on a long polysaccharide and break off one at a time, at a time, at a time. Or you can break it randomly in the middle and then start breaking off those randomly in the middle. Those all work. So this is hydrolysis.